Hello, students. Very good evening. I hope you can hear my voice well. And before we head on to today's class, I would like to know 1 second. Just I'll launch a technical poll. Okay. Yep. So here's a technical poll. So you just have to tell me if the video is clear and my audio is clear, and you can see the background very clearly. And in the YouTube, students, if you have any problem with the voice or any technicalities, please let me know. And it would be very helpful if you just give me a suggestion if you find any technical issues. Okay. All right. Yep. um I'll just wait because I can see that there is a lot of lag between these two channels, so I'll just give that time for it to reach. Okay. So the simple point, I think the YouTube students, you can just put it on the chat box. I will definitely check if there is any technical issues. If not, good enough, we can go ahead to today's class. So in today's class, actually, we have only completed a very little portion of respiration. So it's quite necessary that we understand the core concepts of aerobic respiration before we can answer some questions. Okay. So we'll try to understand these concepts and then we will try to answer some of the questions. So in the last class, we were talking about glycolysis, right? So during our discussion about glycolysis, so what did we talk about it? So glycolysis is one such process that happens anaerobically, correct? So it starts with breakdown of glucose, right? So that is first thing. So what is glycolysis? It's anaerobic first thing. And second thing is it starts with glucose. So we also understood that the stored material in plants is starch, and starch have to be converted into glucose. Once this conversion is complete, now in the cytoplasm of the plant cell is where the glycolysis happens. And remember, it is an oxygenic process. It does not require any help of oxygen. Very important point. Okay. Now, we understood that it was not one-step process. It requires so many different enzymes to help the breakdown of glucose, correct? So the glucose was finally, was broken down into two molecules of pyruvate. So here is where we were ending the process, correct? It resulted in two molecules of pyruvate. So now, this is the point where we take it further. Now, once we have two molecules of pyruvate, we understood that this utilization of pyruvate can happen in three important steps: 1, 2, and 3. So what is that? This pyruvate undergoes either anaerobic respiration or aerobic respiration, correct? So let's talk about anaerobic respiration. If it undergoes anaerobic respiration, usually we call it as fermentation process, which we use it for commercial purposes, correct? Where in commercial purpose, like for example, production of alcohol, and anaerobic microorganisms, usually or anaerobic organisms, what they do is they convert this pyruvate into ethanol, okay, or alcohol. I'll write it as alcohol because it is easy to remember. Okay. So first one is alcohol. Okay. So this happens in yeast or it happens in anaerobic bacteria, etc. And the second point is in eukaryotic organisms, like complex organisms like us, you take human beings. So I told you that imagine that you're working out and you're in the gym and then you are like performing a workout continuously, and you feel muscle fatigue. Muscle fatigue is when your muscle gets tired. When do your muscle get tired? When your muscle is not getting enough oxygen. So when your muscle is not getting enough oxygen, automatically it goes into an anaerobic process where the pyruvate is converted into lactic acid, correct, right? So lactic acid. So these two are anaerobic, okay? These two are anaerobic. I'll write it as, uh, sorry, I don't have enough space, I feel. Okay, I'll write it here, okay? So they are anaerobic processes, right? So, and finally, we have an aerobic process that happens in majority of the organisms with sufficient supply of oxygen, which we call it as aerobic respiration, where the pyruvate is going under a complex processes called as oxidative phosphorylation, finally to give us ATP molecules, correct? So I will write it as aerobic process, okay? Aerobic. Finally, we have aerobic respiration. Remember, the conversion of pyruvate into alcohol or the conversion of pyruvate into lactic acid. Now you might say that, okay, there is a process called as anaerobic respiration. And what is the yield? It means we want some output out of it, right? So if you see this anaerobic process, we understood that it will only give us 7% of the total energy. That means it is not giving the high energy yield. It is a low energy yield process. Whereas in aerobic respiration, the energy yield, that means the output of the energy is very, very high. Okay. So today it's our job to understand the aerobic respiration. Whereas while we were studying the anaerobic pathway, let's just quite revise that so that we don't break the chain of understanding. Okay. So what happened? Let's say in anaerobic pathway, we have fermentation process, and I will give you names of two important enzymes that are very, very crucial for your NEET examination, and I hope you remember that, okay? And I will put the hopes on it. So now we have glucose. We know that glucose will be broken down to pyruvic acid or pyruvate through glycolysis. That is done. Now let's say that we are having alcohol fermentation, let's say. So in alcohol fermentation, there is a very important enzyme called as alcohol dehydrogenase. Okay. So this alcohol dehydrogenase, what it will do is it will oxidize NAD plus to NADH2. And by the way, it helps in conversion of pyruvic acid to ethanol and carbon dioxide. So the carbon dioxide evolution will take place with respect to fermentation process. Whereas if you look at the another type of anaerobic respiration that we talked about, where the pyruvic acid is converted into lactic acid, here what happens is there is another enzyme called as lactic acid dehydrogenase, or it is called as lactate dehydrogenase, we will call it as. So here also what is happening, the pyruvic acid NAD is converted into NADH2 by reducing it, and then we have the production of lactic acid. Remember these two enzymes, alcohol dehydrogenase and lactate dehydrogenase. Okay. So these are very, very important enzymes that you have to remember when it comes to anaerobic respiration, people. Okay. So now let's go for the aerobic respiration, which is very, very important for our many questions that comes in NEET. So in aerobic respiration, what happens? First of all, the aerobic respiration itself is a very complex process that involves various steps. Okay. But we can summarize this step into simpler forms. So what is happening here? We have the pyruvate that is produced in our cytoplasm. Okay. So pyruvate was, let's say that we have a plant cell. Okay. We have a plant cell. And in the plant cell, we have a mitochondria. Okay. We have mitochondria. I'll just write it like this so that you understand. We have mitochondria. And in the cytoplasm, what is happening here? We have the production of our pyruvate. Okay. So this pyruvate that is produced in our cytoplasm has to be transported in our mitochondria. That's the first step that is happening here. So once the pyruvate gets transported into our mitochondria, in our mitochondria, we know that we have the outer membrane, and the inner membrane forms numerous finger-like foldings, or it forms discontinuous foldings, we can tell. So this discontinuous foldings will create something called as crista, correct, right? Mitochondrial cristae. So once it has produced that, now we can see that within this matrix region of the mitochondria, what happens is the pyruvate undergoes certain changes. Okay, there has to be some interconversions that has to happen. So the pyruvate will be converted into oxaloacetic with combination with oxaloacetic acid. It gives rise to citrate and isocitrate and alpha ketobutyrate. Eventually, what happens by the end of this reaction, it will result in the formation of NADH. Okay, so NADH. And now what happens is the major process that happens in aerobic respiration is after you convert this pyruvate into different substrate, we end up with energy molecules such as NADH and FADH2. Okay, we'll talk about it. So these NADH and FADH2 are nothing but energy molecules. There are some bonds within this which carry that energy. So they will enter something called as EMP pathway. Okay, or sorry, they will enter a pathway called as electron transport chain after your Krebs cycle. So that is also called as oxidative phosphorylation. So here, a proton gradient is used for the manufacture or the production of ATP. So I know that it is, kind of a confusing pathway, or it is like mixed pathway in your head, but we can just simplify it going one by one. Okay. Now, where we have to start? We have to start from glycolysis. Yes, the glycolysis has happened in the cytoplasm that has resulted in pyruvate. And the pyruvate that is produced because of the glycolysis is being transported to the cytoplasm. And once it is, it is being transported to the mitochondria. Once it has reached the mitochondria, now what happens is it enters a process called as Krebs cycle. Okay, it enters a process called as Krebs cycle or TCA cycle, where I told you where the pyruvate undergoes conversion of different substrates and eventually to give us our energy molecules. First, we will discuss about the TCA cycle, and then we'll go about oxidative phosphorylation. Okay. So to summarize this entire reaction, what would be happening in our mitochondrial matrix is the pyruvic acid that we have will undergo a process called as oxidative decarboxylation, we call it as. Okay. So what is oxidative decarboxylation? That name oxidative decarboxylation, we'll understand it. So pyruvic acid is converted into acetyl-CoA, which is the final product. Okay. So during this conversion of pyruvic acid into acetyl-CoA, we get some important byproducts. So to that, there are various complex enzymes and byproducts that play an important role to accomplish this process. We have pyruvic acid, which was produced. It combines with a CoA, okay? And then with the help of NADH plus in the presence of magnesium ion and a very important enzyme called as pyruvate dehydrogenase. Very, very important to remember this enzyme, because who conducts the process of oxidative decarboxylation of pyruvic acid? If the question is asked, your answer should be pyruvate dehydrogenase is an enzyme. Okay. So pyruvic acid in the presence of CoA plus NAD plus, what do we get? In the presence of magnesium ion and pyruvic dehydrogenase, eventual product would be acetyl-CoA and then carbon dioxide as a byproduct of our aerobic respiration. And then look here, NADH and H plus. Okay, very, very important. Okay. Now what happens? We have acetyl-CoA that eventually enters our Krebs cycle. Can we go and see what is happening in the Krebs cycle? Fine. So if you look at the process of Krebs cycle, right? See, hold on. So this is the Krebs cycle. That is a TCA cycle. Now what you have to understand is, for the pyruvate to enter the Krebs cycle, it can't enter into a Krebs cycle as a pyruvate, but it has to be converted into acetyl-CoA. Correct, right? That's what we understood. Now you can see that our pyruvate has how many? It has 3 carbon atoms. Correct, right? So the entire process is involved in removing this carbon dioxide, carbon atoms to give rise to our carbon dioxide and water. Okay. And the rest of the process, that is pyruvate, in the presence of CoA, NAD plus, and then pyruvate dehydrogenase, okay, will give us what? Acetyl-CoA. Okay, it's a multi-step process where the pyruvate dehydrogenase complex will participate in. Okay. Now, once we have this irreversible reaction where we have the acetyl-CoA, now our acetyl-CoA is a base product, I can tell. It is a starting material for our TCA cycle. Understood? So let's see what happens with respect to our TCA cycle and how the acetyl-CoA is utilized. Fine. Now, coming to our TCA cycle, okay, it is also called as tricarboxylic acid cycle. There is a reason for that. Or it is also called as Krebs cycle, who discovered this or who mapped this entire pathway. Okay. So let's begin with what happens to this acetyl-CoA, which is in the mitochondrial matrix. Okay, which is in the mitochondrial matrix. What happens to that? So acetyl-CoA, which is in the mitochondrial matrix, it combines with our oxaloacetate. Oxaloacetate will be there in the mitochondrial matrix. We'll understand where we get this oxaloacetate. This oxaloacetate combines with our acetyl-CoA, and this reaction, what we call it as combining, is called as condensation process. Okay. So during this condensation process, oxaloacetate combines with our acetyl-CoA, okay, in the presence of an enzyme called as citrate synthetase. Okay. Citrate synthetase will catalyze this reaction where acetyl-CoA will be eventually converted into citrate, okay, or citric acid. Once we have the citrate, what happens is it is isomerized. Okay, the citrate will be isomerized into isocitrate. Okay, we can call it as here. So the process is intermediate. There is an intermediate process where citrate is converted into cis-aconitate, and cis-aconitate is converted into isocitrate. But if you look at your textbook, the textbook says that your citrate is converted into isocitrate. See, this is an isomerization reaction very clearly where there is just rearrangement of your atoms that are present in your citrate structure. Okay. So let's get back again. We had acetyl-CoA. Acetyl-CoA combined with our oxaloacetate, and this combining process, what we call it as? We call it as condensation reaction. And this condensation reaction was catalyzed by an enzyme called as citrate synthetase. Okay, with the use of one more water molecule. So eventually, what do we get? We get citrate. And the citrate, what we get is finally converted into our isocitrate. And this reaction is an isomerization reaction. Okay. So where citrate is converted into isocitrate. Once we have isocitrate, what you can understand is here is where the important steps that starts to occur. See here, we have 3 carbon atoms in the structure of our isocitrate. During the next following process, what happens is there is a systematic removal of this carbon dioxide from the structure. So let's see how it happens. The isocitrate, okay, with the application of NAD. See, NAD will enter into this cycle where NAD is reduced to NADH. Very, very important disease. Okay. NAD is reduced to NADH. And in the presence of our isocitrate dehydrogenase, okay, what happens is a carbon is removed along with the oxygen where CO2 comes out. So what does it result in? The isocitrate, when a carbon dioxide molecule is removed from its structure, it will give us alpha ketoglutarate. Now, if you see the structure of alpha ketoglutarate, what do we have? We have only 2 carbon atoms, and the rest, the third carbon atom is what you're seeing is a carboxylic group. Okay. So now what happens? We have alpha ketoglutarate. Once we have alpha ketoglutarate, again NAD plus will come into this cycle. Again, it goes into a reduction process where NAD plus is reduced to NADH and then H plus. So here, the enzyme that helps us is alpha glutarate dehydrogenase, okay, where alpha ketoglutarate will be converted into succinyl-CoA. Very, very important byproduct that we have here that is called as succinyl-CoA. Again, here what happens? One more carbon dioxide molecule is removed. Okay. So the succinyl-CoA along with the CoA, what it gives is, see, we have succinyl-CoA and then we have our enzyme called as succinyl-CoA synthetase. Now, till here, we have systematically removed 2 carbon dioxide molecules. But our aim is we have to regenerate one of the important byproducts that is necessary for the TCA cycle to continue. What is that? We need to get back to oxaloacetate. For the regeneration of our oxaloacetate, what happens is the succinyl-CoA is converted into succinate by our succinyl-CoA succinyl-CoA synthetase. Once we have succinate, the succinate will be converted into following products called as succinate will be converted into fumarate. Fumarate will be converted into malate. So these intermediate processes are also supported by our important enzymes called as succinate dehydrogenase. Now, when we have conversion of succinyl-CoA to succinate, it just happens with an enzymatic process. Okay. Here, for this enzyme to act, it requires energy. And that energy comes through the application of GT. Okay. So now GDP enters into our cycle where we get the GDP becomes GTP by the end of this process, which is an energy molecule. Okay. And this is coupled by utilization of an ATP molecule. ATP will be converted into ADP, and this phosphate will be utilized for the production of our GTP. I know that this will be kind of complex for you, but I will repeat this, but just try to listen to this entire process. Okay. So succinyl-CoA was converted into succinate with the enzyme called as succinyl-CoA synthetase. So once we have this succinate, during this process, we got one molecule of GTP. Now, once we have succinate, you have to just observe this process. The succinate, as it converted into fumarate, again, one more important molecule comes into this play that is called as FAD. The FAD is reduced to FADH2, very, very important byproduct, that is FADH2. So FADH2, and then we have NADH, a very, very important energy molecules that is responsible for the production of energy. Okay. So these two byproducts are the key ingredients out of our TCA cycle. Got it, people? Fine. Now, what is happening? We have our succinate. During the process of succinate that is being converted into fumarate, which is coupled by this enzyme called as succinate dehydrogenase, we have FAD that is reduced to FADH2. Once we have a fumarate, the fumarase enzyme, okay, with the addition of one water molecule, will convert it into malate. And malate, eventually, with the help of an enzyme called as malate dehydrogenase, and one more place where NAD plus enters, and it is reduced to NADH2 to eventually give rise to our oxaloacetate, which is a very, very important part. Okay. Now, when you observe this cycle, what you understand is by the end of this cycle, I would be getting, okay, 1, 2, 3. At 3 places, I will be getting NADH2. Correct, right? At 3 places, I'll be getting NADH2, okay, which is very, very important. NADH, not NADH2, NADH. Very, very important that you have to observe. And then I have one place, right? I have one place where I am getting FADH2, correct? And then by the end of the cycle, I'll be getting 2 molecules of ATP. And then there is a release of carbon dioxide that is happening with respect to this cycle. Okay. So now, if you have to revise this entire cycle, let's go again. So first, where does it begin? The entire cycle begins with oxidative decarboxylation of pyruvate. The pyruvate directly cannot participate in the TCA cycle, so it has to be oxidatively decarboxylase. Okay, it has to do that. So how does it happen? This is a complex process which requires so many different intermediates. So it starts with pyruvate, availability of pyruvate in the mitochondria. So for that, an enzyme called as pyruvate dehydrogenase participates, okay, along with CoA and NAD plus, eventually to give us acetyl-CoA. Understood? So this acetyl-CoA enters our citric acid cycle, or it is also called as TCA cycle. Okay. Now, how does it happen? First, the base material that we require for the beginning of this cycle is called as oxaloacetate. Oxaloacetate combines with our, acetyl-CoA in the presence of an enzyme called as citrate synthetase to give us citrate. So citrate is the starting byproduct, I can tell. Citrate is converted into isocitrate by isomerization reaction. Isocitrate is converted into alpha ketoglutarate. In this process, we have NAD plus converted into NADH. Okay, very important. Now, alpha ketoglutarate is again, okay, converted into succinyl-CoA. During this process also, one more NADH is converted, NAD plus is converted into NADH by the reduction process. Okay, again, one more carbon dioxide molecule is evolved. Now, once we have our succinyl-CoA, this succinyl-CoA has to enter the regeneration process. So the succinyl-CoA is converted into succinate, Here is where we get the GTP. Okay. So once we have the succinate, succinate undergoes intermediate conversion of fumarate and malate. So during the conversion of succinate into fumarate, that is where our FADH2 is produced. Okay, because it goes into reduction process. So succinate is converted into fumarate. There we get FADH2. Finally, the fumarate that we get undergoes the conversion into malate and malate into oxaloacetate or oxaloacetic acid. Now, this malate conversion into oxaloacetic acid requires again reduction process through oxidation process through by reducing NADH, NAD plus. So NAD plus is converted into NADH by reduction. When it is reduced, malate is oxidized to oxaloacetic acid, and that is how the completion of our citric acid pathway or TCA cycle is done. Okay, if you look at here, what happens? Where does the TCA cycle takes place? It takes place in the mitochondrial matrix. One pyruvate molecule produces what are the byproducts that we get? We get 4 NADH by the end of the cycle and 1 FADH2 and then one molecule of ATP. Whereas if we compare to the output of one glucose molecule, you just have to double it. We end up with 8 molecules of NADH2, 2 molecules of FADH2, and 2 molecules of ATP are produced by the end of citric acid pathway. Okay, people? So let's go for the next one. Now, once we have the byproducts of our TCA cycle, okay, just give me a second. Yeah. I hope you guys understood TCA cycle. And I can go for the electron transport chain, right? If you have any doubts, you can post it in the chat box. Clear? So now, what is this electron transport chain? Electron transport chain is also called as oxidative phosphorylation. To make it very simple for you, what happens in here in the whole process is, by the time we have the pyruvate, we know that the pyruvate is converted into acetyl-CoA, and the acetyl-CoA entered into another cycle called as Krebs cycle, where eventually we had, FADH2 and NADH. Correct, right? We had FADH2 and NADH. So this FADH2 and NADH, what are they? They are carrying energy in their chemical bonds. Now, what we can do is, by oxidatively reducing this particular molecules, we can get electrons. So the energy stored in these molecules can be broken down. When it is broken down, it releases electrons, and the release of electrons can be used for the manufacture of ATP. Now you must also try to link the two processes. You must also try to link the two processes. Now remember, when we had a discussion about how the ATP synthesis happens through chemiosmotic pathway that we discussed in the chapter photosynthesis. So in chemiosmotic pathway, what did we understand? Our thylakoids, they can synthesize ATP molecules by taking advantage of a proton gradient. So we understood that there is a gradient. Gradient means a difference in the proton charge. So this proton gradient is used as an advantage, and it is used for the synthesis of ATP. The same exact process happens even in the electron transport chain. So the electron transport chain occurs in the mitochondrial membranes. Remember this. Our mitochondria has an outer membrane and it has an inner membrane. Between this outer membrane and the inner membrane, there is perimitochondrial space. The gradient that is created between the perimitochondrial space and the inner membrane of the mitochondria is what drives the entire process. Now, how does this happen is a question mark. And it's a very interesting cycle to understand. And let's take one by one to understand this process. Now, most importantly, before we understand this process, the significant thing that you have to remember throughout this is what has what, or what is present where. Okay. So the electron transport chain is conducted by 5 different complexes. And let us name this complex. It's named, these complexes are named as complex 1, complex 2, complex 3, complex 4, and complex 5. Now, with these complexes, they participate in series way where they transport electrons from one complex to another complex with the help of so many electron transport carriers that are present during this process. Okay. Now, many times the questions are asked, what is present in complex 4? What is present in complex 3? What is this mobile electron carrier, etc., etc. So that is why I would like to just take a moment for you to give this tip, or I would ask you to copy this down so that it would be very, very helpful when it comes specifically to this chapter. I have seen questions mostly concentrated on the composition of what is present in each complexes. So that is why I'm specifically stressing on concentrating on this. So let's look at this. So if I talk about complex 1, what is present in complex 1 is a very important enzyme called as NADH dehydrogenase. Very, very important. Okay. And it is also having some of the prosthetic groups such as FMN and FES, which helps this process. Okay. Next one. What is present in complex 2? Again, I told you. See, if I tell you about complex 1, you have to just have it in your fingertips that complex 1 as NADH dehydrogenase. Very important. Okay. When I talk about complex 2, what does it have? It has FADH2 dehydrogenase. Okay. Or it is also called as succinate dehydrogenase. Sometimes you get confused with succinate dehydrogenase and FADH2 dehydrogenase. People think that it is two different things. No, it is called by different names, but it is the same enzyme, I can tell. Okay. Complex 2 as FADH2 dehydrogenase, and then it also has FES similar to that of your complex 1, and one more important electron carrier called as ubiquinone. Very, very important. Okay. It also has ubiquinone. So complex 1 has NADH, complex 2 has FADH2 dehydrogenase, and it also has ubiquinone. Okay. Next one. Let's go for complex 3. What do complex 3 have? As in questions coming from complex 3 also, just look at it. Complex 3 has cytochrome B6. Yeah, cytochrome B, I can tell. It has cytochrome BC, and then it has cytochrome B, it has cytochrome C, it has FES, and also ubiquinone. Okay. So when you talk about complex 3, you have to talk about cytochrome. What kind of cytochromes do we have? Cytochrome BC, cytochrome B, cytochrome C, and then also ubiquinone or FES. Clear? When it comes to complex 4, now here also we have cytochrome, okay, and then C oxidase cytochrome here. When it comes to complex 4, remember this cytochrome A1 and cytochrome A3. Okay, so why cytochrome A1 and cytochrome A3 are important? Because they have something called as copper centers, which is very, very significant. Okay. And finally, we have complex 5. Complex 5 is very easy to understand because you just have to remember the structure of ATP synthetase, which has F0 and F1 particles. F0 and F1 particles, we have a little bit idea because we talked about this same thing in our chemiosmotic pathway. So let us just rest that particular thing, having the confidence that we know about it. Okay, that we'll discuss in the end process also. So where does everything start? So you have to remember, first we will take consideration of complex 1, and then we have complex 2. Okay. Now what is happening in complex 1 and complex 2? In complex 1 and complex 2, what is happening is we have our oxidation of our NADH into, sorry, we have a reduction of our NADH into NAD plus. And then in complex 2, what is happening? FADH2 is again oxidized, is reduced to FAD. Okay. So what is happening in complex 1 and complex 2? In complex 1, with the help of NADH dehydrogenase, NADH plus H plus is converted into NAD plus. Okay. Next, what is happening in complex 2? In complex 2, succinyl dehydrogenase or FADH2 dehydrogenase, what it is doing is it is converting FADH2 to FAD. So during this process, what does it result in? It results in production of our electrons. So whatever these electrons are produced by the reduction of these two important components. So what happens here? Here, the electrons that are released by this process, okay, these electrons that are released by this process is given up, okay, they give up these electrons to this electron carrier, which is present in the inner membrane of the mitochondria. So who is this electron acceptor? This electron acceptor is called as ubiquinone. Understand this. So once this ubiquinone receives these electrons from complex 1 as well as complex 2, now this ubiquinone carries this electron from complex 1 and complex 2 and it goes and gives to our complex 3. Now this complex 3, giving the electrons to complex 3 do not happen very easily. Why? Because once I have this electrons accepted, that means there has to be some chemical conversion of the ubiquinone. Ubiquinone is converted into ubiquinol. So once the conversion of ubiquinone is done to ubiquinol, now what happens is it releases electrons to its complex 3. Once the complex 3 receives this electron, now you can see that the complex 3 receives the electron through cytochrome B, and the cytochrome B transfers, it transfers to cytochrome C through FES, okay? Just like passing down these electrons through these chemical conversions. So once we have the electrons reaching to cytochrome C, now the cytochrome C is a mobile electron transporter. Remember these people, very, very, very important. Now this is present across our, just outside the inner membrane, okay? Now, once this cytochrome C, which is a mobile electron carrier, it carries the electrons from the cytochrome, complex 3 all the way till the complex 4. Now, the cytochrome C, which is a mobile electron carrier, it gives out its electron finally to our complex 4. Our electron acceptor in our complex 4 is our copper ions. Very, very important. Who receives the electrons from our mobile electron carrier such as cytochrome C is our copper ions. Once our copper ion receives this electron, it goes through our, I told you that they have two important components. What are those? Cytochrome A1 and cytochrome A3. So it passes through cytochrome A1, cytochrome A3. Again, it eventually goes to copper ions. Here, what happens, see? Finally, the electrons are eventually eventually reaches this position, and here it is coupled. The electrons are coupled with the oxygen. Okay, the electrons are coupled with oxygen, where oxygen finally acts as hydrogen acceptor, and this is where oxygen and hydrogen combines eventually to give rise to water. Now, why all this electron transport chain happened? And what is happening here? Now, once it is reached here, now the next process is there is finally, here is where the small difference happens. During this process of electron transport from complex 1 to complex 1, complex 2 to complex 3, and 3 to 4, okay, and finally it is coupled for the release of water. Now, here in each stage, what you can see is while the electrons are transported, the protons are pumped, okay? See, the NADH, which is present in the matrix, it comes to the inner membrane. See, here is where the hydrogen ions, okay? Hydrogen ions from the matrix, it passes through the inner membrane of mitochondria, and finally it goes into the inner mitochondrial space or perimitochondrial space. You can see that as the electrons were transported here in the complex 1, the electrons were pumped into the from matrix to our inner mitochondrial space, and complex 3 also pumps the hydrogen atoms from the matrix to the inner mitochondrial space. And again in the complex 4 also what is happening? It is pumping the hydrogen from the matrix to the inner mitochondrial space. So each stage, see, complex 4 pumps 4 hydrogen, complex 3 pumps 4 hydrogen, complex 3 pumps 4 hydrogen, complex 4 pumps 2 hydrogen. So the hydrogen concentration in the inner membrane space is increasing very drastically. Now we have high proton concentration in the inner mitochondrial space. Now these protons enter complex 5. Okay. While it enters the complex 5, this is a very interesting phase because we already know, and it becomes very, very easy. When we look at our complex 5, it is nothing but it has some of the particles of our ATP synthetase. Our ATP synthetase has two particles. One is F naught particle and F1 particle. F naught particle, okay, is present as a pump. Okay, it looks like a pump which is embedded in the inner mitochondrial membrane. Now it receives this hydrogen ions and it vigorously pumps these hydrogen ions from the inner mitochondrial space towards the matrix. As the hydrogen ions are pumped towards the inner mitochondrial space towards the matrix, the pumping action, okay, as the hydrogen, the protons enter into this, that will lead to conformation changes in the F1. So once this F1 goes into this conformational changes, F1 has a very important part of our ATP synthetase where it starts adding phosphate to our ADP. So this ADP plus phosphate will be converted into ATP in our F naught particle of our complex 5. Eventually, it gives rise to our ATP. Okay. Now, you must, it's so easy and it's so interesting to understand this whole process of how the ATP synthesis will take place. Correct, right? So if you did not understand, can we just go and revisit this particular, just give me a second. I'll just erase this. Yeah. Now we can just revisit what we just studied. Correct, right? So where were we? We finished our TCA cycle and gave rise to NADH and FADH2. And this NADH and FADH2 are energy containing molecules. And somehow we can use this FADH2 and NADH for the production of ATP. How does this happen? Through oxidative phosphorylation. For that, we have to go from matrix to the membranes of mitochondria. We know that our mitochondria has two membranes, outer membrane, inner membrane, correct, right? We have outer membrane, we have inner membrane, and between this, we have perimitochondrial space. That means there is a space between these two membranes, okay? Now, the difference in the proton concentration between the perimitochondrial space and the inner membrane is where everything matters, okay? So what happens here? We have 5 complexes. Complex 1 has NADH, dehydrogenase. Complex 2 has FADH2 dehydrogenase. Complex 3 has cytochrome B, cytochrome BC, cytochrome B, cytochrome C, and then it has ubiquinone. And then complex 4 has the same cytochrome, but it has copper ions as well as A1 and A3 cytochrome. Okay. And then finally, we go to the complex 5, which is an ADP synthetase. Clear? Now, what happens? Complex 1, FADH is reduced to FAD. Okay, sorry, NADH is reduced to NAD, and then that gives rise to what? Electrons. So these electrons are carried by the complex 1, and they give up to the ubiquinone. Ubiquinone receives these electrons from complex 1. Done. Now let's go to the complex 2. What happens here? NADH2 is reduced to, FADH2 is reduced to FAD by the succinyl dehydrogenase in the matrix. So that happens. What does it give rise to? It gives electrons. So what does it do? Again, it goes and gives to the ubiquinone. Where is ubiquinone present? In the inner membrane of the mitochondria. Clear? Now this ubiquinone receives the electron from complex 1 and complex 2. Now the ubiquinone have to go and give it to the complex 3. Now this doesn't happen simply. Ubiquinone is oxidized to ubiquinol. So when it is oxidized, it gives up its electrons to our complex 3. Now the complex 3 takes these electrons and then it transports this electron to our complex 4 through a mobile electron carrier, which we call it as cytochrome C. Cytochrome C is mobile. That means it can move from one complex to the another complex, so which is present in the inner mitochondrial space. So this cytochrome C, which is a mobile carrier, carries the electron from complex 3 to the complex 4. Complex 4 finally receives these electrons, and this receiving happens through the copper ions. So finally, the released electrons are coupled with the oxygen atom, finally where oxygen acts as a hydrogen acceptor. So it removes the hydrogen that is present in the matrix basically. So the hydrogen combines with oxygen to give rise to water, and this water volume is no less. Around 20% of the water volume that is present in your body is because of this process. That means you can understand how much water gets released during your aerobic respiration, right? So once we have the water released through the complex 4, now what is happening in the entire process? In the entire process, by the way, as we are transporting the electrons, the hydrogen is being moved from matrix to the inner mitochondrial space. Where does the movement of the hydrogen atoms has taken place? In a complex 1, it moves the 4 hydrogen atoms from the matrix to the inner mitochondrial space. Same thing happens in the complex 2, where when we reduced our FADH2 to FAD, it moved 4 hydrogen atoms from the matrix to our inner mitochondrial space. So this is one place, this is the second place. And finally, when we are coupling our electrons to our oxygen, so there also 2 hydrogen atoms got released. So now the hydrogen concentration is very, very high in our inner mitochondrial matrix compared to that of our inner mitochondrial space compared to that of our matrix. So this difference is called as a proton gradient. Now the complex 5 takes advantages of this gradient. Now it starts pumping this hydrogen atoms, which is very high in our inner mitochondrial space, towards our matrix. Okay, it goes back. Okay. Now, as it is pumped, it enters to 2 important particles of our complex 5. One is called as F0, other one is called as F1. Now, F1 is basically a proton pump where it accepts the hydrogens and it channelizes towards the F0. Now, F0 is a very important, F1 is a very important part of our ATP synthetase, where it starts converting as the hydrogen is pumped. It goes through conformational changes that leads to conversion of ADP into ATP. And finally, what do we have throughout this entire process that we studied from glycolysis? Production of ATP. Clear, people? Yeah. So I would recommend highly to remember what is present in each complex. So important. Okay. So now, if we look at the whole process and if we summarize, what did we get out of this? Okay. So ATP that is synthesized in organisms are majorly, vast 90% of the ATP comes through our electron carriers such as NADH and FADH2. Okay. And the glycolysis process gives rise to very little amount of ATP. Okay. So how many ATP did we produce in glycolysis? 4 ATP. Out of the 2 went in investment, and finally the yield was only 2 ATP. So if you take the entire process of aerobic respiration, we will get around 38 ATP molecules, out of which 4 are only produced by our glycolysis, and then rest of it is produced by our the aerobic respiration. Okay. Chalo. Now another important concept that we have to study is, what is the balance or respiratory balance sheet, we call it as. Like that means in glycolysis, how much yield we get. In oxidative decarboxylation, how much yield we get. In Krebs cycle, how much yield we get. So totally, what is happening here? So the glucose, if we have to see the entire process, the glucose breakdown takes place through catabolism, and eventually it is given 36 ATP. Okay. And then it gives rise to 6 molecules of water and 6 molecules of carbon dioxide. Okay, let's see. The glycolysis process will yield us 2 ATP molecules. As I said, out of 4, 2 goes for investment and 2 remains as a, profit, right? So these 2 molecules of ATP, 2 molecules of NADH2 will give us 8 ATP. Now you might be confused. How did we get 8 ATP? Each molecule of our NADH2, okay, can give us 3 ATP. Okay, each molecule of NADH2 can give us 3 molecules of ATP. Whereas FADH2 can give only 2. Okay, got it? FADH2 can only give 2. Fine. Now what is happening? This is the yield, ATP. 8 ATP will get through glycolysis. Now, do not get confused, people. When the glycolysis process happens, we are getting how much? Only 2. While the glycolysis process is complete, only 2. By the end of aerobic respiration, if you just look at the bigger picture, then it is giving us to 8 ATP molecules. Remember this. So that is what you have to remember. By the end of aerobic respiration, what is the yield that we get through glycolysis? If this is the question, what is your answer? 8 ATP molecules. Got it? By the end of glycolysis, what is the yield of ATP we get? What is your answer? It should be 2. Okay. What is the Total yield, if they get, you should get 4. Now, how is it even possible? Well, like, what are you talking? If you just ask me this question, remember this. While we finish glycolysis, it resulted in 2 ATP, 2 NADH2, and I told you that it enters into our Krebs cycle, which energizes the further reactions. Correct, right? Didn't I tell you that? Exactly. So now, by the end of aerobic respiration, we get 8 ATP molecules. People, by the end of glycolysis, it is only 2. Got it? Clear? 2 ATP and 2 NADH2 is the right answer. Now, coming to oxidative decarboxylation. What is happening in oxidative decarboxylation when the pyruvate is converted into acetyl-CoA? So there what happened, NAD was reduced to NADH2. We saw that since we have 2 pyruvate molecules, that means it will release 2 NADH2. Correct, right? So when you have 2 NADH2, each NADH2 can give us 3 ATP molecules. 3 2s are 6. So that will result in 6 ATP molecules. Done. Next, coming to Krebs cycle, right? That is a TCA cycle. What is happening in TCA cycle? We get 2 molecules of GTP, which is again coupled in the to form ATP molecules. So we get 2 ATP molecules there. And we know that 3 places is where the NADH2 is being utilized. Correct, right? I told 3 spots we have NADH2. So 3 into 2, 6, because we have 2 pyruvate molecules out of 1 glucose. So we get 6 NADH2 when it is converted. 6 3s are 18. So we get 18 ATP molecule. And then 2 molecules of FADH2 we get, and then you know that each molecule of FADH2 can give us 2 ATP molecules. So, and then it gets like 4 ATP molecules when it is coupled. We get our total 24 ATP out of the whole Krebs cycle. Clear? Now, this is a very, very important thing that you have to remember. Whenever there is a respiratory balance sheet, that by the end of aerobic respiration, if they ask what is the yield of glycolysis, remember it is 8. By the end of aerobic respiration, what is the yield of oxidative decarboxylation? Remember that it is 6. By the end of aerobic respiration, what is the yield of Krebs cycle? You should tell it is 24 ATP molecules. So by the end of aerobic respiration in eukaryotes or in eukaryotes, we get 36 ATP molecules, and 2 of them will be spent in the beginning of glycolysis. We know that. Whereas when you talk about energy production in prokaryotes, you should tell it is 38 ATP molecules. Remember this difference? Aerobic respiration in prokaryotes will give us 38 ATP, and then aerobic respiration in eukaryotes, okay, will give us 36 ATP molecules. Where does the 2 ATP molecules are spent in eukaryotes? By transportation. The transportation process happens from your cytoplasm to our mitochondria. For that, we utilize 2 molecules of ATP. So what is being transported from cytoplasm to mitochondria? NADH, correct? So that is where we lose 2 of the ATPs. Okay, clear? Chalo. So I think we are out of time. Now I will just give the room for you. If you have any doubts, very, very important concept, students. So if you have any doubts, can you please let me know? And if you don't have any doubts, well and good. Okay. So now we can easily go for solving the questions because we have only two cycles that can be done in a very short time. Okay, no problem. Students from YouTube, take your freedom to just put your doubts in the chat box. Okay. I'll just type it here. Get done. Please, it's very, very simple. Just take one concept at a time and break it down. And if you feel that, oh, okay, I'm not understanding, there is, a problem in getting the concepts, don't worry. Next class, you keep appearing and then make sure that you just post your doubt in the classroom. And please don't carry the doubts for a very longer period of time. Why? Because I know that next new concept comes and the new concepts appears difficult and you leave behind the old concept. Okay. So that shouldn't happen. That is why I ask you, whenever you feel that, okay, today I studied it and tomorrow I'm not getting it. So don't worry. You just post it out, and definitely we'll look at the YouTube as well as the charts that is posted during the classroom. Okay, and then we will definitely help you clear those doubts. These are some of the important topics, that is plant physiology and human physiology, and we should take no negligence in these topics, because in plus two comes and everything becomes volatile in the plus one. But remember, people, once you go through the concepts, somewhere in the back of your mind, the concept still remains intact, even though you don't remember everything systematically. Definitely, it remains in the back of your head. All it requires is a revisiting of these topics as we go down and write our face our examinations, right? So that is why it is highly essential to just target our doubts at a right time. Okay. Chalo. Thank you very much for attending this session. I'll see you in the next class, students, and we'll solve some questions then.